Welcome to the online tutorial about cells, Chapter 7, pages 182 to 207. This is Ms. Ziron. In order to understand better what we know about cells, we need to take a step in time, back to 1664 when Robert Hooke took a piece of cork and took a look at it under the microscope and discovered little compartments. He named those compartments cells. Now, Anton van Leeuwenhoek is credited for viewing the first living things using his own particle microscope that he designed. He took samples from pond water and also from his teeth. It is important to understand that the work of many scientists over a long period of time is what leads us to new and exciting discoveries in science, like, for example, Schleiden, Schwann, and Virchow's work, which led us to the cell theory. So the cell theory includes three principles. It says all living things are made of one or more cells. Cells are the basic unit of life, and cells come from pre-existing cells. You're going to find a lot of theories and very few laws in biology. What they have in common is that they are based on hypotheses, they're backed up by a lot of data, and they are pretty much accepted by the scientific community. The difference between them is that the theory is an explanation of an observed phenomenon and the law is a description of it. An example of it would be the motion of the planets, which is described by Kepler's law, but it really does not provide an explanation for their movement. The discovery of cells would have never been possible without the invention of the microscope. The compound light microscope is the one we have in class. With it, you can see living organisms, for example, in samples of pond water like algae and microscopic worms. You can also see tissues of plants and also chick cells. You really cannot tell any other organelles beside the nucleus. The TEM microscope is much powerful. You can actually see the little parts that make up cells, in this case the mitochondria, and the chloroplast, which are organelles with specific functions. The SEM microscope will let you take three-dimensional images. However, just like the TEM, the specimens are dead. Let's take a look at general information about cells. We know that all cells have a plasma cell membrane, they have genetic material inside them, and they all need to break bonds to release energy. Cells can make up unicellular organisms, and those are made of one cell, or they can actually make multicellular, which are composed of many cells. In multicellular organisms, cells take on different shapes and functions through cell differentiation. This happens the minute that the egg is fertilized by the sperm. The zygote, or the fertilized egg, will grow, and eventually their cells will change into blood cells, muscle cells, and other types of cells as well. All your cells have the same DNA, but in order for them to change and have different jobs, they have to turn on and turn off different genes. This slide gives you an idea of how cells are different from one another in animals. This happens as well in plant cells. We have different types of cells for different types of jobs. When we talk about cells, we can actually divide them into two groups. We have prokaryotes versus eukaryotes. Now, prokaryotes, pro means before, kary means nucleus, so it's before the nucleus, and these are the simple cells. These guys lack a nucleus. They're simple and very small compared to the eukaryotes. They're made of just one cell. They do contain DNA, but it's in a circular manner, and they divide very simply as well. An example of it is a bacteria. On the other hand, eukaryotes have a nucleus. They're very complex. They can be one or made of many cells, and examples of them are algae, yeast, plants, and animals. All prokaryotes have a cell membrane, a cell wall, ribosomes, circular DNA, but no nucleus. In eukaryotes, we can also find cell membrane, ribosomes, DNA, but they do have a nucleus, and they do have complex membrane-bound organelles. An example of those would be protistas or amoebas found in pond water, fungi like molds, plants, and animals. Examples of eukaryotes also include parameciums, which are found in pond water, just like amoebas. So the biggest difference between prokaryotes and eukaryotes is the size of the cell. Prokaryotes are smaller, and the presence of the nucleus in eukaryotic cells. The similarities are that they do contain genetic material, ribosomes, and they are similar in their basic metabolism. Now let's take a look at cell membrane, which all cells have. The cell membrane is sometimes called the fluid mosaic model. The reason for that is that the phospholipids and proteins can move around freely, and the mosaic is because if you see the cell membrane from the top, you can see patterns produced by the scattered proteins. The main function of the plasma membrane is to control what goes in and out of the cell. However, it is also involved in cell-to-cell recognition. It is a protective barrier. It helps the cell keep its shape, and it also attaches to other cells to create tissue. In addition, it is the binding site for enzymes. Now, all cells are surrounded by a plasma membrane. Now remember, it has two layers, just like a sandwich. It's also made of many phospholipid units. Now, many macromolecules are essential components of the cell membrane. We have the phospholipids, the proteins that help with transport, cholesterol that maintains its flexibility, and the carbohydrates that help with cell-to-cell recognition. The phospholipid is divided into two parts, the head, which is water-loving, and the tail, which is water-fearing. You will always find the tail part of it tucked inside the cell membrane. Remember, the phospholipid can move sideways to allow small molecules to enter, such as oxygen and carbon dioxide. Now, these are just a few other diagrams that will help you understand the structure of a phospholipid. The cell membrane also contains proteins. We have peripheral proteins, which are attached to the surface or to the sides, and we have the transport proteins, which are also called integral proteins, and they help transport materials through the cell membrane. The glycoproteins are just another type of protein that have the carbohydrate tails attached to them. Remember, the carbohydrate tails are the ones that help with cell-to-cell recognition. Selective permeability is a property of the cell membrane, and what it means is that some molecules will be able to cross through the cell membranes and others won't. Now let's take a look at the types of transports that the cell membrane has. Transports can be divided into two main categories. We have passive transport that require no energy, and they move molecules from high to low concentrations. Active transport, on the other hand, will require the cell to spend energy. So passive transport is described as going down the concentration gradient. You don't need any energy to go with the flow. On the other hand, active transport is going against the concentration gradient, so you do need energy to go against the flow. There's three types of passive transports. We have diffusion, which is used by oxygen molecules and carbon dioxide. We then have facilitated diffusion, which involves the use of a transport protein. And then we have osmosis, which is a type of diffusion that involves water molecules. Now remember, diffusion doesn't need any type of energy. It goes from a high concentration to a low concentration. Osmosis is the diffusion of water molecules, and we see that this movement of water has an effect on plants and animal cells. The types of solutions that involve the movement of water will include hypotonic, isotonic, and hypertonic. So the movement of water through the cell membrane is dependent on the solution that's surrounding the cell. Let's take, for example, the plant cells. They will lose water if they're put in a hypertonic solution. They will gain and lose water at the same rate if they're put in an isotonic solution, and they will gain water if they're put in a hypotonic solution. By the way, hypotonic is the best condition for plant cells since they don't have a skeleton to rely on. On the other hand, animal cells reach homeostasis when they're put in isotonic solution. They will shrink in a hypertonic solution, and they will burst open if put in a hypotonic solution. So just as a review, a solute is something that you dissolve in a solvent, and together they form a solution. Now let's take a look at active transport. Now active transport moves molecules from a low concentration to a high concentration. Notice that there's only two of these molecules outside, and there's 5 molecules on the other side. This is possible with the use of a carrier protein and also ATP. Other types of active transport will involve the cell membrane, and this is endocytosis and exocytosis. And as the diagram shows, it involves the cell membrane surrounding nutrients that it needs, or a vesicle in the case of exocytosis merging with the cell membrane to release waste or other unnecessary materials. This chart represents the summary of the transports that we have just discussed. Now let's take a quick view at the levels of life and how it's organized. Atoms make molecules, molecules make organelles, organelles make cells, cells make tissues, tissues will make organs, and they will make organ systems, and finally everything will be put together in an organism. The parts of the eukaryotic cells are as follows. We have many types of organelles with very different jobs. The main difference, and we're talking about eukaryotic cells, is that plant cells will have cell walls, central vacuoles, and chloroplast. On the other hand, animals do have centrioles, which are not found in plants, and they also have lysosomes, which are very rare in plants. The function of the organelles include the nucleus, which contains the DNA or genetic material, and it's also the control center of the cell membrane, which is where transport takes place, the ribosome, which creates proteins, the ER, which is called the endoplasmic reticulum, which helps assemble and transport proteins, the Golgi will package them and export them, the lysosome, which contains enzymes that help break down parts that the cell doesn't need anymore, the cell wall that helps with the rigid and the support in plants, the central vacuole, which helps contain and store nutrients and water in plants, the chloroplast, which is compared to the solar panel. It holds green pigments and it helps the plant capture the light energy and convert it into food, the mitochondria, which helps make ATP molecules from the food that we eat, and the cytoplasm, which is a jelly-like material that holds the organelles. This is just a quick diagram of the animal cell, which is a eukaryote. In animal cells, remember, centrioles are basically found in only animal cells. Another picture of the plant cell. And in plant cells, we have the cell wall, the central vacuole, and the chloroplast that are unique to the plant cell. And this is the end of the online tutorial on cells. I hope I have clarified many of your questions. Please let me know if you have any more. Thank you and see you in class. Bye!